So first of all, thanks to the organizers for putting this together and also for bringing me here. It's very nice being back in India, and I'm really enjoying this meeting so far. So what I wanted to tell you today, I was actually not so sure about until very recently, and I did change some of the things I included in the talk yesterday. So let's see, this somewhat vague title, Traffic on Filaments, Motors in Difficult Conditions. I'm not 100% sure how much that really fits what I'm going to tell you. I think to some extent it does. I wanted to start by showing you this movie again. I think you have seen this before. And actually last time in Anatoly's talk, and he asked you what is wrong in this movie. Who remembers what's wrong in this movie? Yes, one person in the back. It's just a cartoon, yes. Yes, there should be more things bombarding this cargo. Anything else? In the very back, yes. I couldn't hear it, but it's probably... Okay, there's only one motor. Yes, that's another thing that is most likely wrong. So if you want to... Yes, it looks a little bit too organized. Yeah. Okay, so if you want to see a more realistic movie, you should go to YouTube and watch the movie from the Hogenrad lab, where you see a molecular motor stepping through the streets of what I guess is probably Amsterdam or some other city. And where all these challenges that a motor faces during a normal day in its life are nicely explained. Here is a list of the things that I thought might be challenges that you could not see in this movie, and that does to some extent correspond to what all of you just said. To some extent, this corresponds to things that I will or will not talk about in this talk. So the first issue is stochasticity. So everything should be much more irregular. Everything should be fluctuating a lot more because we are in this range where thermal fluctuations have a very important role. Then there should be forces. The motor not only pulls, but the motor can pull against the force, and the force may either be due to some external thing. The cargo may be attached somewhere and has to be pulled off. I will show you an example in the second half of this talk where this opposing force is magnetic. But then, of course, force can also be due to other motors. That's something we heard about quite a bit yesterday, where you have different types of motors moving together, and either if they have different speeds or because they differ in some other way, or in the extreme case, because they pull in different directions, you will have forces that one motor exerts and the other motor experiences. And then there's this whole issue of crowding that initially I thought I was talking about more in this talk, but now I decided to skip it. And we heard a little bit about crowding in the talk by Sandhya 3 days ago. And crowding, I think in that case, can mean two things. Either you can have obstacles directly on the track of the motor, or you can have just a crowded cytoplasm around, which would be obstacles for the cargo that is pulled through, and would be obstacles for the motor if the motor has to find the filament to start moving in the first place. And the last thing I wanted to include here is that the track itself could also be dynamic, and you'll see a little bit of that at the end of the talk. Okay, so what I want to tell you about in this talk is two things that are more or less unrelated. The first part will be relatively short. I will briefly tell you about some recent work that we did on the tug-of-war between teams of motors that pull in opposite directions. And then in the larger part of the talk, I will tell you about a dynamic filament. So in that case, the polymerization of the filament itself is what generates motion and forces. And this is specifically a filament that is found in bacteria, namely a filament made of a protein called mamk, which is a bacterial actin-related protein that moves magnetic particles in magnetotactic bacteria. Okay, these are two projects that I at some point thought I would include but didn't. So if you're interested either in roadblocks in exclusion processes or in DNA binding in crowded conditions, then we should talk about this in some break. Okay. The first topic I wanted to talk about is the tug-of-war between teams of motors that pull into opposite directions. And I will not tell you all the work that we have done 10 years ago, but rather only focus on some very recent thing. And this very recent work was done by an undergraduate student in my group, Omar Munoz, who came basically to me, I want to work theoretically on molecular motors. Can you give me a project? And so he ended up doing something on this tug-of-war model. This is something we have already seen in several talks. There are these bidirectional motion where cargos in cell move in one direction for a while, and then after some time they move in the opposite direction. So typically you have this back and forth motion, which could be very fast, like in this example here, where the turning around happens on a second scale, or it could be very long runs, like here, where it moves into one direction for several seconds and only later turns around. Anyway, you have a cargo that rather than moving from one place to a defined destination in a straightforward manner, makes this back and forth bidirectional motion. This, by the way, is data from Steve from quite a while ago. There is another example down here, a bit more recent, well, not that recent either anymore. And you also see these two different types. So you have this one particle here that moves back and forth relatively quickly, and here you have another one that shows this rather long run in one direction and then followed by a run in the opposite direction. Okay, you have also heard in previous talks that we know quite a lot about the single motors. They are very well characterized with respect to the velocities at which they move, the forces they generate, their chemomechanical cycles, and so on. So the paused state, what that is, well, you'd have to ask Steve, what is this paused state? But there are many ways how you could generate a paused state. It could be motors pulling in both directions at the same time. It could be linked to something else and be stuck transiently. It could be that both motors have stopped pulling because they detached. It could be, there are many reasons, many mechanisms how you could generate a pause. I don't know which one that is. Okay, so I just said the single motors are typically rather well characterized, and so one question was, how does one couple them together? And if you have several together, what coordinates this motion? So is this turning around an effect of some biochemical signaling, or is this turning around just something that happens randomly because the motors are coupled mechanically and there is a tug-of-war and sometimes one side wins and sometimes the other side wins? So that has been a discussion at the time, maybe 10 years ago or so. And at that time, we got into this and we said, let's take this tug-of-war idea serious and let's see how far we can get with that. And I only described this very qualitatively and very briefly. So the idea is you have two teams of motors, they pull into opposite directions, just like you have two teams of people pulling some cargo in opposite directions. And how could that lead to some bidirectional motion rather than just to a stall? The idea that we had at the time is that what you need is mechanical forces, and these mechanical forces, these you get for free because the motors pull, but then what they have to do is they have to trigger some kind of instability where motors under force unbind more easily, and then this can trigger a cascade where all the motors of one team unbind, and then the other motor team will basically win and pull the cargo in its direction. So something like this will happen. One team will basically fall off. The motors in the cell don't have gravity, so they will not fall to the ground, and the other team will pull this cargo rather quickly in one direction. Okay, so since then, I think there has been quite some evidence that these tug-of-wars do happen. There has also been quite some evidence of data that is not consistent with just this mechanical work. So one example, maybe the nicest demonstration of a tug-of-war was this work of Roop Malik's group in 2009, where they showed that during this turning around, in that case of endosomes, these endosomes elongate, providing evidence that they are really pulled on in both directions at the same time. This is another study by Adam Hendricks and others, where they showed that if they analyze quantitatively the motion of cargoes, then this, for neuronal vesicles, what they showed there was quantitatively consistent with what the tug-of-war model predicted. There are also a few observations that show that something is not just mechanics, which is also not so surprising, because after all, these are things happening in cells, and we know that there is signaling and there is biochemical regulation. So why should it not affect molecular motors? So one example is here from our organizer, Ambarish, who showed that long pauses in the motion of lipid droplets were not consistent with a tug-of-war, even in his more stochastic version compared to our model that then called the mean field model. Another example is this one from the Holzbauer lab, where they showed that a regulator called GIP1 could induce very long runs that went into just one direction. So these basically switched between a kinesin and a dynein and then induced motion that was straight in one direction over large distances. And another example that I like is this one by Leidl et al., also in lipid droplets, where they showed that when you pull a lipid droplet off from a filament and put it back on, it tends to move in the same direction as before more often than you expect. If everything is mechanics, whenever you pull it off, the memory should be gone, but there is some memory, so this must be something that's beyond just mechanical pulling. Does this mean that mechanics doesn't play a role? Well, I don't think so. The simplest reason for that is that you cannot get rid of mechanics. As soon as you have two types of motors and they pull, of course, the force will be there. So in general, I would expect that we have mechanics, which is kind of a generic mechanism for coordination, and you have biochemistry, which is, of course, much more specific. So the one thing I wanted to show you that my student Omar did very recently is that we try to come up with models where we have some signaling included in a very abstracted and toy model type fashion. So this should look different. Anyway, you shouldn't see those states. You should see these three. So these three states in the center is basically our absolutely minimal model for bidirectional motion. The idea is we have motion either in the plus direction or in the minus direction. There is switching between them, and then also the cargo can unbind and not be connected to a filament at all. So these are these three states. And then what he did is that he included some additional states here, another state where motion is in the plus direction, another state where motion is in the minus direction. The idea is that some signaling mechanism locks the direction of motion. So this is the idea. We can go in plus direction and lose the ability for switching. For example, because the minus motors are turned off in some way. So and then they can also unbind from this state. So that's something we allow here. And one thing you see if you look at some pieces of trajectory in this model is that already these look very diverse. You have these trajectories where you have this rapid change of direction between plus and minus, which is basically given by switching between here and here. But then in addition, you have trajectories where you have very long runs that go just in one direction, which is of course induced by going into these states where the direction is locked in. So what this type of toy model would tell you is that you should generally expect that you have rare but very long runs, which is seen in many of the experiments. Typically, you would then get at least these double exponential distributions of the run length or times. This is time. And the other thing which you get from this state is that when you unbind a motor from here, if you take it off from this state, in this state, so if you let it rebind to a filament, it should have a higher probability of staying again in the plus state. And you see this is if you look at these two curves here, the upper curve, so the probability to go into plus direction again after unbinding and rebinding is increased for some time. Of course, if you keep it off too long, then these transitions here will happen. You will lose the memory, but for some transient period, there will be a memory of the direction in which the motor has gone before. Well, the idea is that you have some modification of the system going from this state to this. It could be that, say, the dynein motor is somehow inactivated. It could be that the kinesin motor is modified and activated so it would always beat the dynein motors. We don't really say this in the model. This is a simulation. This is in the simulation. We don't specify what the mechanism is. Yeah, I would expect that this is very specific. I think that the situation that you have long runs, rare, very long runs, that's relatively common. This is seen in many systems. And even in this system of Adam Hendricks, where he showed that the model is consistent with just this mechanical tug-of-war, they said that very rarely there are some runs that are longer than what we expect. Yes, but this you can also get from other reasons. So this alone doesn't prove much. But this is very common, and I think the fact that you have very long runs that are rare, that's a general observation. Okay, so I wanted to show you briefly one idea how one could generate this with a somewhat more microscopic model. So we went back to the tug-of-war model that we had studied 10 years ago. And this is one small piece of data as a reminder. So in this model, basically, we describe the state of the system by the number of plus and minus motors that link a cargo to the filament. And typically, we have one of two situations. Either, so this is a probability distribution of these states, either you have mostly one type of motor bound or both types of motor bound with equal probability. Yes, this is the 4-4 state. So here, typically, the most likely state is the one where both motors are bound in equal numbers, namely all of them. This is 4 against 4, and then typically there will be not much motion. In this case, no motion over this time scale. But if we have parameters differently, and that is mostly this ratio of stall force and detachment force that we have talked about yesterday, we heard in the talk by Manoj, then this distribution becomes bimodal, and you have typically either one team bound or the other, but not both of them together. And that will then lead to this bidirectional motion, where basically you switch from this state to this state in a stochastic fashion. If we now include activation of motors, so motors can individually be activated or inactivated, and in the simplest case we do it stochastically, then with the otherwise exactly the same parameters, you now are actually very similar. So this difference between the two different regimes of the original tug-of-war, the pure mechanics, completely disappears, and you get bidirectional motion more or less independently of the parameters, because what you do now is basically you switch by this activation from situations where the plus motors are dominant over the minus motors to those where the minus motors are dominant just by numbers of active motors. So this does provide a mechanism for this. Of course, what is important now is what are the rates of these activations and inactivations, because this will basically be a distribution over long times, and over short times it could still look more like this, depending on where you are. Okay, so this was some perspective I wanted to give on this combination of regulation and mechanics. I think there's still a lot to be done there, but eventually this is a very important issue. I think we also identified this in one of the discussion sessions. How do we combine the mechanical interaction of these motors with the biochemical signaling that will certainly also play a role there? Yes. I will switch now, yes, so ask now. Okay, I think these are two things that I said. One thing is, if you have the memory, yes, you will get these long runs. Well, in my model, I don't have the memory all the time. I just have it a fraction of the time, so you get some long runs, but you also get short runs with rapid changes of direction. That's when you don't have the memory. So basically, if you have or have not the memory would depend on at which time point you grab the motor and unbind and rebind it. The other thing I said is that if you have a mechanical tug-of-war, and mechanics is all you have, then getting bidirectional motion will depend on specific choices of parameters, while if you have activation, inactivation, you will generally get bidirectional motion, and the parameters don't matter that much. In particular, in this case, I mean, basically you don't see a difference. There's some small difference, but it's not visible. So these are two points. Okay, let me now switch to another topic, and this is a filament that is related to actin in magnetotactic bacteria, and I assume many of you have not heard about magnetotactic bacteria before. Who has heard about magnetotactic bacteria? 2, 3, 4, well, a few, more than I thought. Okay, so this shows some motion of magnetotactic bacteria. This is a droplet of water that we look on from the top, and you see bacteria swimming all to the right because there is a magnetic field that points to the right, and they accumulate here at the surface of the droplet. And to convince you, something seems to be wrong. This is the same movie again. That's not what I wanted to show you. I wanted to show you this movie. So now you have bacteria that have accumulated here at the right. Something is not correct. This one, no, okay. Now we turn the magnetic field. You see the magnetic field is now turned to the left, and you can make them all swim in the opposite direction. Okay, so these bacteria can be steered with an external magnetic field. And if you want to see some more fancy movements, you can go to YouTube and just type in dancing magnetotactic bacteria, and you see some much nicer. Don't do this now. No, you can do it now. Fine. Okay, so how do they do this? They do this because they have a special organelle called the magnetosome chain, which is this structure here. And that is basically a compass needle in these cells. And with this, they can orient in a magnetic field in their natural habitat, this magnetic field would be the magnetic field of the Earth. One zooms into that a little bit, one can see the structure. So you see there are these particles. These are colored in red. So this is a reconstruction from cryo-EM. You see the particles. These are iron oxide crystals, so magnetic nanoparticles. They are wrapped in vesicles. You see the yellow membrane around these particles, and they are attached to this green structure here, which is a cytoskeletal filament that is made from a protein called mamk, which is a bacterial actin-type protein. What is important, the magnetism of these particles is permanent, so these are really small, permanent magnets. They are magnetically single-domain particles, which means that they have the highest possible magnetic moment for the material. Okay, so the simple picture of these bacteria, how they move in magnetic fields, is that you have passive alignment, so basically just a compass needle that orients the bacteria, and then they swim with the help of flagella, just like other bacteria swim with flagella as well. One point I want to stress is they are not attracted by the magnet, not under the typical conditions, because typically, certainly for the magnetic field of the Earth and also for the magnetic fields used in experiments, you have a homogeneous field. So there is no magnetic field gradient that would attract these bacteria. So the only thing that the field does is align these bacteria. So they have to swim. If they don't swim, they will not move along the magnetic field. So this is alignment and swimming in a simulation. I will not talk much about this simulation, but I thought I'd show you the movie because I think it's very nice. You have a flagellum that rotates, and this pushes the bacterium forward, and you see that it does slowly align with the direction of the magnetic field, and then eventually it will swim out of the screen. Okay, so what these bacteria need is a compass needle, and one structural requirement for this organelle to function as a compass needle is that it should be reasonably straight. Because if it's straight, then all the magnetic moments of the particles point in the same direction, and you have the maximal total magnetic moment, which should give you the best alignment with the magnetic field. The one thing we were thinking about, what is actually the bending rigidity of such a system? And this bending rigidity should have two contributions. One is due to the filament, should be similar to the bending rigidity of actin, hopefully. Nobody has measured it, but let's assume it's similar to the bending rigidity of actin. But then in addition, there is a magnetic contribution, because when you bend this chain, then you perturb the orientation, the alignment of the magnets. So that should give you an additional contribution, and that's something that one can calculate, and one ends up with a surprisingly simple expression for that is basically given by epsilon, which is the magnetic interaction between two neighboring particles that are aligned, the distance, and then a numerical factor. This theta of 3, I think, is 3 point something, so this is all very close to 1. If one puts in the numbers, one can calculate this bending rigidity, because this epsilon mostly depends on the magnetic moment of the particles, which is known. We can also convert this into a persistence length, and the persistence length that one gets from that is something like 3 to 4 microns, and this is comparable to the length of these cells. This is only the magnet, these 3 to 4 microns. This is only the magnet. If you take the filament to be like actin, then you get something that's actually higher than this. Persistence length of actin is maybe 10 microns, so somewhat more than that. Okay, I just told you, in principle, we have a chain of magnet that is persistent. Sorry, this was too fast, but anyway. So this should be straight on the scale of a cell. So why do we actually need an actin-like filament there? Well, one thing you see in this movie is that a straight chain of magnets, even though it's stable and has a persistence length, it is only metastable. This you can do in this little experiment. This is actually my daughter doing this little experiment. If you perturb it a little bit, it snaps back, but if you perturb it too much, then this chain of magnets is closing into a ring, which has the lower energy if you go to somewhat more than, I think, 4 particles. Yes. If you have 4 particles, then this ring will be the lowest energy structure. So one thing we think this filament is doing is it stabilizes this chain against closure of the ring, which would, of course, completely destroy the function as a compass needle. We've also recently run some simulations. Let me see what is happening now. So Bahari Kiani, who is a recently graduated PhD student in my group, she performed simulations where she let these particles assemble into structures, and without a filament, actually the filament in the simulation is still there, but the particles don't bind to the filament. You get mostly these ring structures. You also get some of these rings with a tail. Occasionally get chains because the chains are metastable. But so you have quite a large fraction of these ring structures rather than chain structures. While if you allow the particles to bind to this filament, then also in the assembly, what you get is mostly these straight chains. Only occasionally you get these very elongated rings like this one. They don't form these large rings that we see in the simulation. So there are some mutants that form very small rings, but typically they have many small rings that tend to cluster. So it's less ordered than the structures that we get in the simulations. So there's 2 types of these mutants. There are 2 important proteins. One is the mamk protein that makes up the filament. If you remove that, then what you get is mostly some shorter chains. So there must still be something that makes some structure. There's another type of mutant where the mamj protein is removed, and this is a protein that is supposed to link the particles to the filament. So based on the simplest cartoon picture, they should do exactly the same thing, but they don't. So in that case, you get these clusters of particles and small rings. In general, yes, but you still get some magnetotactic motion, because what happens, if you have a strong field, what happens is that these clusters will tend to elongate a little bit, so you still have some net magnetic moment. Yeah. I don't think it has been quantified, but actually that's something that should be done. Okay, so this is one thing. One other thing about the structure of these particles, I should actually move on to filaments. Let me skip this. So if you want to know about this, what we do here is we probe the structure by rotating an external magnetic field, because then we can use the magnetic forces to exert force on these magnetosome filaments. This is done in a simulation. We also have done this together with my collaborator in Potsdam, Damien Fèvre. We have also done this experimentally. And you get some structures like this where you get short chains that are now perpendicular to the direction in which they should be. So what you see is that the chain breaks into smaller chains. But let me move on, because this is a meeting about motors. Let me talk about the active part of these filaments. So this we started some years back. Damien Fèvre, my collaborator, he did experiments where he starved these bacteria for iron. So he kept them in a medium without iron for a long time, and then shifted them to a medium with iron. So they started to make magnetic particles again. I'm not sure you can see this. There are some tiny particles here after about 2 hours after the addition of iron. You see the arrows, and there are some small black dots, which are the particles. over time, these particles grow bigger, and they also align into this nice chain that you've seen before. So the whole process takes about 5-6 hours until you have reformed this magnetosome chain. So the question we asked is, do magnetic interactions play a role? Is it really assembly of magnetic particles due to their magnetic attraction? Or is some specific biological signaling important here? We can ask more specifically, is diffusion plus these magnetic interactions sufficient to form this chain? In the simulation before, we saw that this is the case, but is it true here where we have to form the particles and not have particles originally put in? And then the question is, if yes, what is the role of these mamk and J proteins? And so what we try to do is some kind of mutation in the computer, where we don't knock out specific proteins, but we take out and in specific processes. We do this with a model that combines a Langevin dynamics for the spatial degrees of freedom and the Monte Carlo dynamics for the magnetic degrees of freedom. So this is basically some kind of spin flip. And describe every particle by the position, its size, because the size is growing. So there is some nucleation plus growth, and a magnetic degree of freedom that for the direction of the magnetization. And then we simulate this for about what corresponds to 10 hours of the real dynamics. The nice thing is most of these parameters we can estimate from the experimental data. The only thing that is really unknown is the mobility of these particles. So this is something we vary over a very wide range. And with that, we simulated this case that I just sketched, where we have just diffusion plus the magnetic interactions. And doing that, we find that if the mobility of the particles is low, basically we never get a chain. If the mobility is high, so this is the higher end of the realistic range, then we get at most, let's say, 50-60% of the simulations that end with a single chain, which is the typical case in the experiment. So this is something that usually happens. We end up with two chains that are somewhere. They are typically not in the center of the cell because there is no reason for them to be in the center. And also we see that the overall magnetization comes up much later than what it does in the experiment. So we thought, well, we need something that brings the particles to the center, so we basically assume that there is active transport, and the obvious candidate is the filament itself as transporting these particles towards the center. And if we do that, then of course we can form nice chains in the center. Well, that they're in the center is not surprising because that's basically what we put into the model. But what we see is that it's extremely robust if the mobility of the particles is sufficiently high. What is sufficiently high? Well, this depends on the parameters, but basically there is some threshold above which we get, in most cases, a single chain in these simulations. And this threshold can be related to a competition between bringing the particles to the center and the formation of shorter chains somewhere else in the cell. So if the formation of short chains far away from the center is faster than the transport to the center, then this will not work. But if the transport is sufficiently fast, and it doesn't have to be very fast, so the speed we estimated there is something like per minute, which is relatively slow, then such a mechanism should robustly lead to the formation of one chain. Well, in the model, we basically have a limit that is put in there by hand. In the real cells, I'm not sure what limits that. I mean, it must be a combination of iron uptake and also time, actually, because it takes if you have to form this new, it takes 5 hours, and then after 5 hours, you might, under good condition, that's also when they would divide. So I think under good conditions, it's probably limited by the time until the next cell division. So you form the particles at some constant rate, you divide, and then you have to reform half of the particles, and that would roughly, under conditions where they grow more slowly, I don't know. I'll come to that. Maybe, hopefully. Okay, so let me skip this. There was some evidence that this filament might be dynamic from mutants, but I'll show you in more detail in a moment. There's much nicer, more newer evidence. So I wanted to briefly tell you about cell division. How do these things divide? So electron microscopy showed two things. First of all, these chains are divided. So the cell, when the cell divides, every daughter cell gets half of a chain, roughly. You also see here that the division has this nice kink, which is also what you would do if you have two magnets and you have to separate them. You can try this at home. If you have two magnets, you don't pull them apart like this, but you first bend it and then pull them apart, because then you need less force to do that. And the bacteria seem to do this as well, and that seems to correlate with the fact that the cell wall at the septum grows asymmetrically to induce this kink. Let me skip this. This is a newer picture of cell division. One of my collaborators, Mauricio Toro Nahuelpan, he was in the lab of Dirk Schüler at the University of Bayreuth. Well, at that time, he was actually still in Munich, and the tomography was done at the Max Planck Institute in Martinsried. What you see here is two things. First of all, you see that the cell, this chain is divided, but you also see that directly after division, when you have closed this septum here, that the chain is not in the center of the cell anymore, but it's now at the pole of a cell. So what you have to do is the cell has to solve two problems of positioning this chain. One is before division, the chain has to be in the center, and the other one is after the division, because now the chain is not in the center anymore, and it has to move back to the center. And to get some more insight into this, we analyzed the accuracy of this partitioning. So basically what Mauricio did is he took many of these electron micrographs of cells, and he just counted how many particles are in one daughter cell and how many are in the other. This is a cumulative probability distribution, because that's nice to interpret in this case. Actually, I mostly did that because you need a cumulative distribution to do the statistical test, but it turns out that to see that these curves are different, I don't think you need a statistical test. If you calculate a p-value for this, this is 0 basically, 10 to the minus 30 or so. Anyway, so you see that this is in the wild type bacteria, and you see that the partitioning is very accurate. So only a few % deviation from equal partitioning, you will have most of the probability in here. So actually what we find is that the average deviation from equal partitioning is about 6%, which corresponds to exactly one particle. So typically this is very precise. But he also looked at a mutant, which is a mutant in this mamk protein, and in that case you get this distribution, which is very different, and it's actually very close to the black line, which is what you would expect if the partitioning is completely random. So in this mutant, apparently partitioning is completely random, so this tells us this seems to have something to do with positioning this chain in the center, or at least positioning relative to the point where the septum will be. We also found that this is very well fitted by an exponential, so you could think of this as a balance of drift, active transport to the site where it's partitioned, and a diffusion counteracting it. Okay, and then Mauricio did a lot of experiments where he tried to see the active motion of this filament. I think this is the very nicest one. So in this, he has some mamk that is fluorescent, and the fluorescence is photoactivated. And then you see that at this end you have some red fluorescence, and you see over time this fluorescence does move towards the cell center. And somewhat surprisingly, you also see that a little bit of fluorescence appears at the other pole, and from there also moves to the cell center. So this is what you see more, you see this more nicely here in the chymograph, where you have really motion from both cell poles towards the center of the cell. So this filament really generates some active motion towards the cell center. It's relatively slow, 300 nanometer per minute, slow compared to the molecular motors that we have talked about. It's still fast for these cells. I'll show you in a moment. Yes? So there's a filament through the whole cell. So you have only, basically, you have a subset of subunits that are fluorescent because they are activated here. Some of them certainly diffuse. I think the ones that end up at the other pole of where they're activated, they got there by diffusion. But this sort of extension of the fluorescent area, that I think is incorporation into the filament and polymerization. Anyway, and then he also, I should come to an end now, I'm almost done. He also tracked the particles by a fluorescent label in one of the membrane proteins in these particles, and you see that they also, after division, move from this position near the septum towards a position at the center of the cell. The whole process is much slower, so this takes about 90 minutes. The speed is much less, in this case, 7 nanometers per minute. So this is a really slow process, much slower than the dynamics of the filament that I've shown you on the last slide. And again, in the mutant, this is much slower again. You see that motion towards the cell center. So this is position of the chain as a function of time. Here is slower again by about a factor 10 or so. Most likely from the structure where this mutation is, this mutation affects the hydrolysis. Strongly reduces hydrolysis. And down here you see the same process in this model that we had before, where you can also induce, well, if you look at the division, you get this motion of the two half chains that go towards the cell center in the daughter cells. Okay, so with that, I want to conclude. I think both these systems, this tug-of-war that I have talked about in the beginning and these magnetotactic bacteria, are very nice model systems where we can study this combination of physics and biology. We have forces that are purely physical, either mechanical or magnetic, but they don't act alone, they act together with specific biological processes where you need specific interactions between molecules. There may be many more interesting problems, I think, particularly with these magnetotactic bacteria. But one thing that I don't think I have to stress to a physics audience, but it's very important that you have active processes here to generate the patterns that are seen in these cells. And I said this already, there is, I think, this challenge to understand how these two things work together, the generic physics and the more specific biological signaling. I also put up my collaborators briefly. So I mentioned already Omar Munoz, who has done this recent tug-of-war work, which of course has started long ago together with Reinhard Lipowski and Melanie Müller. But then in the magnetotactic bacteria work, what I've shown was mostly theoretical work by Bahari Kiani and experimental work done on this growth of the chains by Damien Fèvre, my collaborator in Potsdam, and on the cell division by Mauricio Torona-Welpan and Dirk Schüler in Bayreuth. Thank you very much for your attention.
See, this coming to the center is somewhat reminiscent of the chromosomes coming to the center of before cell division.
Yes.
But there, of course, we know the forces that have to be balanced from the two sides by the microtubules. So there is some kind of guiding ultimately that they are brought to the center. Here, what is bringing them to the center and how do they know that they have reached center?
I think the answer is I don't know. We know that there is motion from both sides, which also means that these filaments somehow meet in the center, and there must be something, maybe the crossing of filaments with opposite polarity. Who knows? We don't really know what happens there. But of course, it could also be that the center is determined by another process. We know that in bacteria, there are things like min and so on that determine the center. Maybe so there is no min here, but there's this other, I think Colobacter has a MIP protein and something that's related to MIP present in some of these bacteria. So maybe there is a mechanism that determines the center, and then this mechanism interacts both with, not only determines where the septum is formed, but also determines positioning of the cell or controls activity of these filaments, but it's not really known.
Hello. So is the iron inside this cedar pores is mobile in nature? I think you can take out iron from this organelle.
So what they do is they basically collect iron from the solution, from the surroundings, partly with siderophores, partly with pumps in the membrane, but then they basically package the iron in these vesicles. So you have vesicles, I didn't tell you, basically the vesicles are preformed before you have the particles. The iron is pumped in, is transported into these vesicles. You have additional proteins that control the crystallization in there. You probably have a low pH. And these conditions together induce crystallization of the iron oxide in the vesicle.
Yeah. And the peptide portion of this organelle can have some role in determining the length of the chain. I mean the protein fraction. It's bound by a polypeptide cell. Yeah. So any post-translation modification or modification in the protein itself can have some role in determining the length of the chain.
I think the length of the chain would mostly be determined by the number of vesicles. What proteins determine is the size of the crystals inside the individual vesicles and also the shape. So some species of these bacteria have very asymmetric shapes, elongated, or also some that are kind of, they usually call this bullet shape because they have some little tip on one side. Yeah. And these seem to be dependent on the proteins that are in these particles. Post-translation modifications we know nothing about, so might be. I mean, we're happy.
Yeah, why I'm asking, because this crystal is a mixed crystal. So it has two sublattices because it has two types of oxides. So its net magnetic moment from chemistry point of view depends on some surface defects in the crystal as well as the diamagnetic contribution from the peptide cell also.
What do you mean with two sub-lattices?
Because it's Fe3O4 is composed of FeO, is a ferrous oxide, and Fe2O3, it's a ferric oxide. They form Fe3O4. And in nanoregium, they have generally some surface defects, and the extent of defects determine its magnetic moment. Mm-hmm. And
I'm not an expert on the chemistry of these.
No, it's my
the particles are pretty much pure magnetite. So once they have formed, synthetic particles very often are a combination of magnetite and magnemite, which are two different oxides.
No. In general, Fe3,4 is a mixed oxide. So it has two sublattices.
Okay, if you say so, I don't know. You may be right.
Perhaps we can. Yeah, I just had a question about sort of the role of, you may have mentioned this and I missed it, but I'm sort of wondering, sort of in general, why these bacteria magnetotacks?
Yes, that would be another talk. But basically, the more or less accepted idea is that they use this, they usually combine this with aerotaxis, and basically they use the magnetic field to reduce the aerotactic surge. They usually live in micro-oxic conditions, so they use the magnetic field to reduce the aerotactic surge from a three-dimensional one to a one-dimensional one. Some species apparently use it also to completely get rid of the aerotaxis, because the reason for that is that the magnetic field of the Earth has a component downwards. So if you're in a water, there is some micro-oxic zone somewhere in the deeper layers or upper layers of the mud, basically. And if you follow the magnetic field downwards, that will bring you there, even without aerotaxis.
Okay, I think one last question and then we'll finish.
So to go back to the first part of your talk, is there any quantitative estimation of how fat these tails are corresponding to these really long runs as compared to a random?
I mean, there are many such distributions, so you could estimate it from there, and I would expect that it would be different for every system that you look at.
Okay, but experimentally in any particular system?
There is experimental data, but I haven't looked at this, quantifying this.
Okay.
But I would think it's not universal, but it would depend on the specific system. But you have these double exponential distributions in many systems, so you could quantify it.
Okay, let's thank him again and finish.